Good morning. I am Dr. Sanjukta Naik, Assistant Professor in Department of Biochemistry. Good morning all. Today I will be teaching on the topic urea cycle. So first we will start with what is urea cycle. Urea cycle is also known as ornithine cycle, and it is basically a cycle which includes biochemical reactions that generally produces urea and ammonia. Now coming to what is the site of urea cycle. So urea cycle basically takes place in an organ called liver, as liver has almost all the enzymes which is needed to generate ammonia, and this route is mostly present in the periportal hepatocytes. Now urea cycle generally involves 4 stages. The first stage is known as transamination, which is followed by oxidative deamination of glutamate. Then it is followed by ammonia transport, and then followed by the reactions of the urea cycle. So first coming to what is transamination. So transamination is nothing but an interconversion of alpha amino acids and alpha keto acids. So this reaction basically takes place in the cytoplasm. Almost all the amino acids undergo transamination. Only few except lysine, threonine, proline and hydroxyproline generally do not involve in this transamination reaction. So enzymes which are involved in the transamination reactions are AST, that is aspartic transaminase, and alanine transaminase. So all the amino acids are basically converted through transamination into glutamate. Now this glutamate will then undergo the second step, that is the oxidative deamination. And this oxidative deamination basically occurs in the mitochondria. All amino acids are first transaminated to glutamate, followed by they are deaminated. This is a major mechanism of removal of ammonia from amino acids for detoxification and excretion as urea. Finally, by the process of oxidative deamination, ammonia is produced. So the enzyme which is involved in the oxidative deamination, it is glutamate dehydrogenase. So all amino acids first are transaminated to glutamate. Then by the enzyme glutamate dehydrogenase, it is converted to a keto acid and ammonia. So this is how we get the ammonia. This ammonia finally is transported to the liver, where it will be converted to urea. Now the next step is the detoxification of ammonia. So this is like a first line of defense in the body, as ammonia is highly toxic and therefore it should be eliminated or detoxified as when it is formed. Even in very minute quantities, ammonia is very toxic to the body. Intracellular ammonia is immediately trapped by the glutamic acid to form glutamine, especially in the brain cells. Then finally this glutamine is transported to the liver, where the reaction is reversed by the enzyme glutaminase and we get the ammonia. Finally, the ammonia thus generated is immediately detoxified and converted to urea. Now, the urea cycle was given by scientists Hans Krebs and Kurt Henseleit. It was known as ornithine cycle because ornithine is the first member of the reaction. Then 2 nitrogens are generally present in urea. One comes from the ammonia and other from the alpha amino acid of aspartic acid. Now coming to what are the steps of urea cycle. So it involves 5 steps. First is formation of carbamoyl phosphate. Second is formation of citrulline. Third is formation of arginosuccinate. Fourth is formation of arginate. And fifth is formation of urea. So the first step is carbamoyl phosphate synthesis. So the enzyme involved here is CPS1, and this is basically takes place in the mitochondria. So urea cycle basically takes place in 2 parts of the cells. First is the mitochondria, followed by the other part, which is cytoplasm. So the first step takes place in the mitochondria, where ammonia reacts with carbon dioxide to generate carbamyl phosphate. And the enzyme involved here is carbamoyl phosphate synthase 1. This process is basically irreversible. It is a rate limiting step of the urea cycle and generally consumes 2 ATP. CPS1 also requires a cofactor called N-acetyl glutamate to function. Then second step is formation of citrulline. So citrulline is produced by enzyme ornithine transcarbamylase from the carbamoyl phosphate and ornithine. Ornithine is then recycled and utilized in the urea cycle. The next step is the citrulline formed is then transported out into the cytoplasm, and then the other steps of the urea cycle takes place. So the next step is arginosuccinate synthesis. So arginosuccinate is synthesized with the help of enzyme arginosuccinate synthase. It is when citrulline generally combines with aspartate. This step incorporates the second amino group of the urea from the aspartate. This process basically requires the breakdown of ATP into AMP and pyrophosphate. The pyrophosphate is instantly degraded to inorganic phosphate. Now the next step is arginine synthesis. Now another enzyme called arginosuccinase generally cleaves the arginosuccinate to produce arginine and fumarate. Urea is a direct precursor of arginine. Now urea formation. So the last enzyme, which is arginase, it is the last enzyme that cleaves the arginine to produce urea and ornithine. The regenerated ornithine then enters the mitochondria and it is again reused in the urea cycle. So both carbon dioxide and manganese are activated by the arginase. Now coming to, these were the all steps of the urea cycle. Now coming to the energetics of the urea cycle. So the overall reaction we see is 1 ammonia plus carbon dioxide plus aspartate is converted to urea plus fumarate. So in this reaction basically 2 ATP are used in the first reaction. Another ATP is converted to AMP plus pyrophosphate in the third step, which is equivalent to 2 ATPs. So therefore the urea cycle consumes 4 high energy phosphate bonds. Then fumarate, which is formed in the fourth step, it may be converted to malate. When oxidized to oxalic acetate, it produces therefore 1 NADH, which is equivalent to 2.5 ATP. So we produce 2.5 ATPs while we consume 4 ATPs. So the net expenditure in the cycle is only 1.5 high energy phosphate bonds. Now coming to what is the significance of urea cycle. So as we all know that ammonia is very toxic to the body. Even in small quantity, it can cross the blood-brain barrier and it can go and deposit in the brain. And it can lead to a disease called hepatic encephalopathy, which is highly fatal disease. Therefore, it is very required that we excrete out the ammonia immediately from the body. So urea cycle is very significant as it helps in removal of the ammonia from the body. Then urea cycle also serves for the biosynthesis of arginine from the ornithine. Then in the course of urea cycle, fumarate is produced, which takes part in the TCA cycle as well as gluconeogenesis. Now coming to what is the regulation of urea cycle. So regulation of urea cycle can be classified into two. One is long-term regulation. Long-term regulation basically involves regulation in gene expression. Starving animals and very high protein diet patients, this regulation is involved. It increases in the synthesis of enzymes in the urea cycle. Short-term regulation, it basically involves allostatic regulation of a key enzyme, that is the carbamoyl phosphate synthase 1, and this is done by the activation of N-acetyl glutamate. Now coming to what is the urea level in the blood. So the normal plasma level is generally 20 to 40 milligrams per deciliter. Level is increased when renal function is basically impaired. Now what is the normal ammonia level in the blood? Normal level is from 50 to 25 mcg per dl, or it is 10 to 30 micromoles per liter. Now coming to what are the disorders of urea cycle. So any enzyme deficient in the urea cycle can result in hyperammonemia. When there is block in any one step earlier, the ammonia itself is accumulated, which is what severe. Deficiency result of later enzymes can result in less severe and symptoms. Generally, disorder of urea cycle is described as hyperammonemia, encephalopathy and respiratory alkalosis. The symptoms may include vomiting, irritability, lethargy and severe mental retardation. Now coming to how do we classify the disorders of urea cycle. So they are classified, the enzymes involved in this are basically the first enzyme, which is the CPS1, also known as carbamoyl phosphate synthase 1. If this enzyme is deficient, it can lead to hyperammonemia type 1. Then second enzyme, which is ornithine transcarbamylase, if it is deficient, it can lead to hyperammonemia type 2. Then there is another enzyme which is known as ornithine transporter, which helps in transporting the ornithine from the cytoplasm to the mitochondria. If this is deficient, it can lead to a triple X syndrome, which is known as hyperornithemia, hyperammonemia and homocitrullinemia. Then the fourth enzyme, which is arginosuccinate synthetase, if it is deficient, it can lead to citrullinemia or citrullinemia. That is, the citrulline cannot be converted to arginosuccinate. Then the enzyme, if arginosuccinate lyase is deficient, it can lead to arginosuccinate aciduria. And the last enzyme, arginase, if it is deficient, it can lead to hyperargininemia or argininuria. Now what is hepatic encephalopathy? So it is a CNS disease. It is caused when the ammonia crosses the blood-brain barrier and it gets deposited. So it can lead to features like cerebral edema, intracranial hypertension, neuronal dysfunction, and the treatment is basically the diet, or we get lactulose in the treatment. Thank you.